Okay, so I'm turning on the recording. And as always, I will post the shared screen and not the gallery or the chat. So you're welcome to ask questions in the chat. Okay, so what do we mean with metabolism? It's the set of chemical reactions that either breaks down food to give us, to extract energy in the form of ATP. That's called primary metabolism, or more specifically within the primary metabolism, it's the catabolic part, the breaking apart. There's also an anabolic part which builds molecules like proteins and carbohydrates, all the structures that the body needs if the body grows or maintains its structure, or cells divide and you make new cells. So that's also part of metabolism, but won't be our topic today. Okay. The first part of breaking down food happens in the stomach, and there's no expenditure of energy, but there's also no energy that we extract from it. The way it works is that these are all hydrolysis reactions, breaking things down into smaller parts, and they're downhill reactions. They happen on their own. The equilibrium of these reactions is on the side of the products. So breaking down proteins into amino acids works without putting energy in there. And in fact, you can do that in your laundry by just adding some enzymes, and they will break down proteins, making it easier to get rid of protein stains. Same for carbohydrates and fats. Then there's a part of metabolism that we're skipping. So then we're taking the simple sugars, amino acids, and fatty acids and breaking them down further. And these processes are called glycolysis and beta oxidation for carbohydrates and for fats. You might encounter them later in your studies, but I won't talk about them. But what happens is you get from the bigger molecules that have maybe 6 carbons or maybe 4 carbons, you get to one molecule that has 2 carbons and is related to acetic acid. It's basically acetic acid that is linked to a cofactor called CoA. And so that's how we get this acetyl-CoA. So our concern today will be what happens to the acetyl-CoA. And I can give you the upshot, the 2 carbons, you will breathe out as CO2. So the citric acid cycle that we'll discuss first is a redox reaction, one giant redox reaction that turns the organic molecules into CO2. And at the same time, because it's a redox reaction, if something gets oxidized, something else needs to be reduced. It takes so-called electron carriers called NADH and FADH2 and reduces them. So carbons get oxidized in the citric acid cycle, electron carriers get reduced. And then in the second step, that's more interesting for actually capturing the energy. These reduced energy carriers react with oxygen. And that's a downhill process, and that free energy is captured by, at the same time, turning ADP into ATP. And that's the whole purpose of most of the food that we eat as adults, right? We're not growing that much anymore, or at least we're not supposed to. We're just maintaining body weight. So we're not actually bringing in new material. And the metabolism really is just to provide the free energy to be able to synthesize ATP so that we can do all the things our body does to maintain its healthy status. Okay, so that's the overview, a little bit briefer than I had in the video with less slides, but you can watch those videos as well. Okay, I gave you the overview, how ATP is made, where things happen. One of the interesting things I find that I didn't know up front was I always thought, well, the carbon in food reacts with the oxygen in the oxygen we breathe, and then we breathe out CO2. But using up oxygen happens in a different pathway than making the CO2 does, and it's connected with these electron carriers. Okay, first pathway we're going to look at is the citric acid cycle. So again, this is after molecules have been broken down to acetyl-CoA. Things like glucose and other carbohydrates do glycolysis, and fatty acids through beta oxidation. And this actually starts with the molecule that we get when we come in with glucose. So glucose gets broken up into two molecules called pyruvic acid. Just to remind you, these circles, they mean how many carbons are in there. So pyruvic acid has 3 carbons. That gets oxidized to CO2. And the CoA molecule gets attached, and we get our first NADH out of there. So whenever you see NAD plus going to NADH, that's a reduction. It means that 2 electrons are added to this electron carrier, and now we have something that can later on react with oxygen. So we should be happy for our cell every time an NADH is made, because that enables it to make ATP later by reacting that again with oxygen. Okay. This is a cycle, which is kind of weird. It just goes around and around. You think it doesn't do anything, but there's one thing that's coming in here. That's the acetyl-CoA, and there's 2 things coming out of the cycle. That's 2 molecules of CO2. So it actually accomplishes something. But as it does that, these molecules get regenerated. So we react something with this molecule oxaloacetate. Then there's a series of 10 reactions, and we get oxaloacetate back. And then it can react again with the next acetyl-CoA. So if you want to, you can see this whole cycle as one big catalyst that just catalyzes one reaction, which is acetyl-CoA being oxidized to CO2 while we're getting all these reduction, these electron carriers reduced. Again, so now we can look at the reactions one by one. Before we do that, I want to emphasize, of course, you don't have to memorize these or even learn these. I want you to have the tools to look at a chemical reaction in the citric acid cycle and say, what is going on in principle? So is that a redox reaction? Is this a hydrolysis reaction? That's the basic idea. And to know which molecules are what. This is an electron carrier. This is ATP, which is a way of storing free energy for a short time. Okay, I have 3 slides just filled with reactions. The topics I want to discuss are up here. I just have those as my notes. So many of these are redox reactions, and I want to remind you how to figure out that something is a redox reaction. So if you have a carbon, and that carbon was had a bond with hydrogen, and then afterwards it has a bond with oxygen, that's oxidation. And briefly, the way why we're calling that is the polarity of this bond changes, right? The hydrogen is more the carbon is more electronegative than the hydrogen, so this has a bit more electrons on the carbon, and the oxygen is definitely more electronegative. And so we're taking away electrons from the carbon. That's why we call that oxidation, even though we're not really removing it all the way, but we're going to a bond that is more polarized on this side of the oxygen. Okay, so that's one indication that there's an oxidation going on. The other thing is the special role of CO2. So how many bonds with oxygen does CO2 have? Well, all of them, right? Because we have 2 double bonds with oxygen. So that's the most oxidized species of carbon that you can get. So whenever something reacts to form CO2, you know it has to be an oxidation, because nothing else is that oxidized, and CO2 is the most oxidized, and so that's going to be the oxidation part. Okay, so that's one indication. The other indication is if we have these electron carriers, and by now I've shown you 2, or I've briefly mentioned. So when we go from NAD plus to NADH, that's a reduction. Something else has to be oxidized. The other pair we'll see in a second is FAD and FADH2. And again, the one with the hydrogens, the FADH2 is the reduced species. So this is a reduction. And we could look at the details of this molecule, but we could just say Where we see more H's, that's the direction where it's reduced. That's a good rule of thumb. Okay, so that's the business of redox. And you will find that the first reaction is not a redox reaction. I don't see any of these features. Neither is the second one. We're just adding water and taking water away again to isomerize this. But the third one is definitely a redox reaction because we see this electron carrier being reduced and we're seeing CO2 being made. Okay, so as we're going through these, I will point out redox reaction and the ways we can figure out that it indeed is a redox reaction. Next thing I want to remind you of is hydrolysis and condensation. So here's one reaction that is a hydrolysis. We have acetyl-CoA. Let me make this a little bit thicker here and in red so I can show you stuff. So this has the CoA attached to acetic acid. And in the course of the reaction, this CoA gets hydrolyzed. And the one thing I should tell you about hydrolysis in the context of metabolism, always a downhill reaction. So I told you when you hydrolyze proteins to form amino acids, that's downhill. Equilibrium is on the side of hydrolysis. Same here. So this provides some energy for the reaction. Okay, so the first step, we see this hydrolysis, that the CoA goes away. There will be a point where the CoA comes in again. That will cost some energy. We have a molecule with 1, 2, 3, 4 carbons, and here a molecule with 2 carbons, and then we get this citric acid or citrate, which has 6 carbons. That's how we get to the 6 carbons in our carboxylic acid cycle. This reaction, if you look closely, we're just swapping out these 2 groups. And it's actually a set of 2 reactions, but we don't really have to bother about the details. Okay, so in this set of reactions, we got rid of 1 CO2, we oxidized the molecule a bit, and we got a reduced electron carrier. And those are sort of the topics we'll see. And we talked about the cofactor. One brief note about the cofactor, it's a good trick the cell has to attach a big cofactor, this is actually a big molecule, to a small molecule to sort of hold on to it, that it doesn't lose that small molecule, that it doesn't diffuse out of the cell. So acetate might just leave the cell by diffusion, but acetyl-CoA is too big and too charged to do that. Okay, so we went from 4 carbons and 2 carbons to 6 carbons, and then we got rid of a CO2, and then we're down to 5 carbons. And we're going to be done with the citric acid cycle when we're back to 4 and back to oxaloacetate. Okay, trying to get to the next slide. Not so easy. Much going on. Here we go. Delete this. Here we are. So again, 1, 2, 3, 4, 5 carbons. That's our current status here. And in the end, we'll end up with 4 carbons. So we're losing a CO2 somewhere on the way again. Okay, let's look at the first reaction. You can tell me, do you think that is a redox reaction or not? And then hopefully in the chat, you can give me one indication that it is or that it's not. And that's what I mean with being able to sort of make sense of these reactions, not maybe knowing exactly what the molecules are, what alpha-ketoglutarate and succinyl-CoA is, but looking at the reaction products and reaction partners to see what's going on here. So does anybody see some indications that this might be a redox reaction? You can write it in the chat. And While I'm waiting for somebody to say something, I'm preparing a poll here, so we'll do this in a different way. Do you think this is a redox reaction? Yes or no? Let's try it this way, then everybody can participate. Okay, I gave you 30 seconds to answer. 10 people say yes, it's a redox reaction. 3 people say no. So there seems to be like a gut instinct, at least, that there might be a redox reaction going on here. Now I'm challenging the 10 people who said yes to give me their reasoning in the chat. You might have expected this to happen. And as you think about that, maybe write something, which would be nice if I hear something. NAD, yeah. So here's one indication. Part of the reaction is this redox carrier. And good answer. I don't have to say who it was. You know who you are. It goes from NAD plus to NADH. So this is being reduced. And so what we expect is that going from here to here, there's actually multiple products here. There's the CO2 and there's the succinyl CoA. We have oxidation. The amount of carbons decreases, yes, because we're making that CO2. So definitely this carbon that comes out as CO2 is being oxidized because it has the highest degree of oxidation or oxidation state that it can have here. And then if we look closely, this is kind of difficult to see what's what here. If I number the carbons, I think this is carbon 1, 2, 3, 4, and 5. This is a complaint that I have, that they don't write them up in a way that lines up. And I'm not so fast drawing this. So this carbon that used to be a carboxylate group is now a CO2 that got oxidized. This carbon that used to be a ketone or a carbonyl group is now a carboxylate that got oxidized. Carbon 3 and 2 and 1 did not get touched with oxidation. Carbon 1 is kind of more complicated, and that's another feature of this reaction. So as we start with this is just the alpha ketoglutarate, nothing attached, and we end up with these 4 carbons attached to CoA. I told you that attaching CoA is an uphill reaction, and hydrolyzing it gets some energy out of it. The reason we can attach this here and the reaction still goes forward is because all of this is coupled. We're doing all these things at the same time. It's a bit like having a pulley where we have one reaction that pulls down the weight, and then the little weight can get pulled up at the same time. Anyway, so this is a combination of a redox reaction that you now have identified and a condensation reaction where the CoA is attached to this molecule. And that's a way of sort of storing energy. This is an uphill reaction, and we'll see in the next step what will happen. I will not ask whether this is a redox reaction. I will just tell you it's not. We had the CoA attached. Now we have the CoA leaving. That's a hydrolysis. So that's one part that happens. We're hydrolyzing off the CoA, going from succinyl CoA to succinate. So it's the same molecule, just that attached to CoA and not attached to CoA. At the same time, we're making some GTP. We didn't discuss GTP, but it's the same molecule as ATP. There's just something slightly different chemistry on the site that is uninteresting in this case. Anyway, so this is an uphill reaction, and it's driven by the downhill reaction that we're hydrolyzing the succinate. So that's why this works. And that will be the big topic of making ATP, coupled reactions. So let me say a couple of words about that. So putting a phosphate on ATP, on ADP to make ATP is an uphill reaction. It goes against the direction that the equilibrium would go. I said earlier, enzymes can't change the position of the equilibrium. So you can't just put an enzyme in there and say magic, it'll work. But what you can do is couple it to another reaction that wants to go downhill. And when you add those two up and the free energy of that is still positive, negative, then it works. And so that's exactly what happens here. At the expense of hydrolyzing off the CoA, we can put a phosphate on GDP and make some TTP. Okay, that's the principle of coupled reactions. And in some ways, our bodies are one big set of coupled reactions. That's why we can do this stuff that we can do. That's what distinguishes life from non-living material. So all the non-living material basically erodes over time. Mountains get lower, and I guess minerals get washed out and so on. So you get to a state where everything is mixed and kind of boring. But if you have life, then it builds up these structures again. Not mountains, but separates, can have a situation where not everything is mixed, but you have a high concentration of something here and a high concentration of something else there. Okay. So going to the next bit, we were at 4 carbons already. I said when we're at 4 carbons, we're done, but we're not at the right molecule yet. But so I can assure you that this doesn't go on forever. This is oxaloacetate, sorry. Let me get my pen back. Nope, sorry. Let's try this one more time. It's a pen. Okay, that was our starting point, right? So we're getting back to the starting point. So this is the last slide of reactions. Okay, it's all 4 carbons. So we can't find the redox reactions by looking at CO2 being made. But if I call this reaction A, and this reaction B, and this reaction C, can you tell me which ones of those are redox reactions and which ones are not? And I'm going to look at the chat, see if somebody has an idea. Okay, so the chat says C. I'm assuming that means C is a redox reaction, and that is totally true. And let's first find the easy way of showing that. Here's an NAD plus that gets reduced to NADH. So there's certainly a reduction going on, and if there's a reduction going on, there should also be an oxidation there. If we look at the 2 molecules, malate and oxaloate, they both have a carboxylate group on the top and the bottom, so no changes there. They have a CH2, but here this has an alcohol group or a hydroxyl group, whereas this has a carbonyl group. So that also shows you that this carbon gets oxidized. It had a bond to an oxygen and to a hydrogen. Here has a bond to 2 oxygens, so that's oxidation as well. Okay, so for reactions A and B, what can you say? So you could just write in the chat, yes or no to A and B. Any ideas? If we have another redox reaction here somewhere. Oh, that's it. A is also redox. Here we have the other electron carrier, FAD, gets turned into FADH2. And then this is a bit more difficult, but here we have a carbon that has 2 bonds to hydrogen, and instead now it has 1 bond to hydrogen and the other to carbon. and that also counts as oxidation. Okay, cool. Then let's look at reaction B. We have a double bond here, and then we have a single bond and an OH and an H. We're just adding water. This is not a hydrolysis because it doesn't break the molecule apart. It's an addition across the double bond. Anyway, that's what this is. And there's no electron carriers and there's no ATPs being made. It's just one step in the cycle. Okay, we have a great comment in the chat, which is I agree. That's good. I agree with everything that was being said. Okay, so that's the citric acid cycle. The nitty gritty of it, let me just tell you the big picture again. We have the organic molecules that come from our food being oxidized all the way to CO2 so we can exhale them. At the same time, sorry, we're reducing these electron carriers that we'll need in the next step. Okay. So I could have showed the next slide, I guess, for the overview, because that is, again, the citric acid cycle. And this gives you a little bit about the energy that's available here. So if we were to take acetic acid and burn it with oxygen, which is not happening here, of course, this is a downhill reaction, has a free energy exchange of 873 kilojoule per mole. I should have put a negative sign in front of here because it actually provides free energy. We're not doing that. Instead, we're oxidizing in steps. And so we're getting all these reduced electron carriers that we can use in the next step. And I said at the beginning, and you don't have to remember that, but oxidizing NADH with oxygen gives us minus 220 kilojoule per mole. So this is almost enough for 4 NADH, but not quite enough. And it turns out the FADH is easier to reduce, less energy involved. And that's why we have this combo of 1, 2, 3 NADHs and 1 FADH. Okay. So that was the citric acid cycle. It's the reason why we breathe out CO2, and it provides us a lot of NADH and FADH, which is used in this final redox reaction, which explains why we breathe in oxygen in order to live. So the reaction that actually happens here is, let me box that. We're having NADH react with oxygen, but in a very crazy way. So they're not close together, but the NADH gives off its electrons, and the electrons are involved in a couple of reactions that I should highlight the flow of the electrons in red. They just took the same color. It's not so smart. So the NADH first reacts with an electron carrier called Q to reduce it, and then this Q goes on to reduce something called cytochrome C, and that goes on to reduce something called complex 4. Details are not so important, but these electrons in the end come all together to turn oxygen into water. Okay. So arrows here mean 2 different things. They mean chemical reactions, but they also mean the flow of electrons. Okay, this whole thing is called the electron transport chain. It happens in the mitochondria at the membrane. And again, I have a couple themes that I want to discuss with you that go back to what we studied for the second and the third module. The themes are diffusion, redox reactions, and intermolecular interactions. So the first thing I want to say is that this membrane acts as a barrier. So it doesn't have a gap here. It goes all the way around, right? So we have an inside and an outside. This has a funky name, the matrix. It's a very dense part, protein dense part of the mitochondria. Has a lot of enzymes in there and lots of chemical reactions are going on. Okay, let me tell you the overall gist of this electron transport chain. We have this redox reaction, bringing in electron carriers from the citric acid cycle and oxygen from us breathing, and then of course the bloodstream going all the way to the tissues, into the cells, into the organelles, into the mitochondria. And these two react with each other. That's a downhill reaction. So it wants to go that direction. NADH and oxygen will react to form water and NAD+. And the free energy that's available from that is used to do a process that is not downhill but uphill. And this is pumping these protons across. So there's a lot of arrows here. I should draw these in a different color. So the green arrows are transport of hydrogen ions across the membrane. Okay. Once you transport a couple of H pluses across the membrane, you have more H plus on this side than on this side. So if I let the hydrogen ions move, would they move upwards or would they move downwards here in this picture? Would they go to the inside or the outside? If I just open up the membrane, I have a high concentration of H plus here, and I have a low concentration of H plus in the matrix. In which direction would the H pluses want to flow? You can answer in the chat. Away from the matrix or to the matrix? If we have very few hydrogen ions in the matrix and a lot on the outside, which direction would they want to go? They would want to flow back in, okay? So this direction we call pumping because it goes against the direction of the equilibrium. We're building up what's called a gradient. Let me just write that word down because you might encounter it. So this is called a membrane gradient. It just means that the concentration on one side and the other side is different. So we have a proton gradient. Of course, gradient, oops. Proton refers to the H plus molecules, ions, and gradient refers to having a high concentration here, low concentration there. Okay. The concept of diffusion is that concentrations equalize. So if you have a high concentration in one space and a low in another place, they will mix, and at the end they will be the same. So if these protons could mix between the outside and the matrix, they would do so, but they can't because there's a barrier here, and that goes to the topic of intermolecular forces. This is hydrophobic. We have an ion here. The ion can't just go across. Okay. So this is important that this process takes place as the matrix because the whole point of it is building up this proton gradient. Okay. And then these Roman numerals are proteins that have boring names. They're just called complex 1, complex 2, complex 3, and complex 4. They have fancier long names that describe which reactions they catalyze. Complex 1, 3, and 4 all do sort of the same thing. They catalyze a redox reaction, for instance, NADH giving up the electrons to Q. So that's a downhill reaction, and that powers the pump that transports protons across the membrane. Complex 3 does the same thing. It's just a different redox reaction. It takes the electrons from Q, so it oxidizes Q and it reduces cytochrome C, again, pumping protons. And complex 4, again, similar thing. In this case, the reaction is interesting because that uses up the oxygen we breathe. So that gets oxidized, gets reduced, and cytochrome C gets oxidized. And again, we're pumping protons. The diagram doesn't tell you how many protons we pump. It's actually 4 and 2 and 3 or something like that. It just says protons are being pumped. There's one special case here. This reaction 2 doesn't do any pumping. It just oxidizes the FADH to FAD, and at the same time reduces some of the Q. All of these molecules either come from the citric acid cycle, or there's also other NADH produced by oxidizing fatty acids to acetyl-CoA. Okay, so I checked off all those points. What we didn't say is what is the point of all this? Why we're making a proton gradient? And then we finally get to the production of most of the ATP that are produced in the body. It's a single enzyme. Let me clear drawings and get to the next slide, which is the same slide, just with one thing added. So let me circle that so you can see it. So this part we already saw. That's the electron transport chain. And in the membrane, there's another enzyme called the ATP synthase. It's called ATP synthase because it synthesizes ATP. Putting a phosphate on ADP and making ATP is an uphill reaction. It doesn't work on its own. The equilibrium is on the other side. But in order to accomplish this, we have a coupled reaction again. And this time we're coupling flow of protons, and I guess I was using green for that, so I'll make it in green again. So this is a transport phenomenon, not a chemical reaction. So we had a high concentration of H plus out here. So these protons, they want, or these H pluses, they want to go back into the matrix. And the only way they can is through this ATP synthase. It's a hugely complicated machine with moving parts, fascinating stuff, but we're just going to show it as this pear-shaped blob. As the protons are allowed to flow back into the matrix, the free energy available from that is used to make ATP. Okay. So they're not talking directly to each other, but they're talking via this proton gradient. But that's the purpose of doing all of these reactions, the about 20 different chemical reactions I showed you, is just to make ATP. Unfortunately for us, this works pretty well. And other organisms have been relying on this. I should say this is not only in humans. Any living organism has the same type of metabolism, not always in mitochondria, but the same idea of an electron transport chain and oxidizing and reducing and making ATP. That's universal to all forms of life. Okay. So this is a quick run through how organisms make ATP, and it links it to getting food and getting oxygen into the cells to run all these reactions. We didn't take any time to say what happens to all the ATP, but I briefly said what we do with it. So muscle action, things going on in the brain, building molecules. So when proteins are made from amino acids, you need ATP. So all of these steps that are uphill require ATP, and this is where it's coming from. Okay, so on the next slide, I think I'm showing the grand scheme again. So we looked at the details of single enzymes and molecules, and now we can go back to the big picture. So what happens in the body is, and this I should say is the animal body, or I guess also everything except for photosynthetic plants rely on this mechanism only to make ATP. And I'll say something about plants in a second. Okay, so we have all these foods that we find and grow and make and buy. That gets broken down into the monomers, into the simple building blocks. They get oxidized to acetyl-CoA, which fuels the citric acid cycle. So in the citric acid cycle, these get oxidized, and at the same time, electron carriers get reduced. That's the function of the citric acid cycle. And again, this is why the CO2 is produced that we breathe out. And then I talked about electron transport chain and ATP synthesis. We can also group them together and call the whole thing oxidative phosphorylation, which means that this is a process that makes ATP. That's what the phosphorylation wants to say, and oxidative because we're using oxygen to oxidize these electron carriers. So that's electron transfer chain, and ATP synthase then uses these and oxygen. So really, we should have O2 going in there. That's where we need O2 to make ATP. So that's a brief overview of our metabolism. So I said I wanted to say something about plants. So I told you it takes free energy to make these. And they basically come from plants that we have this free energy. And the trick that the plants have is to take the free energy from the sunlight using photosynthesis, turning CO2 and water into these molecules like carbohydrates that are the source of all the free energy that the animals are using. Okay, so you can always sort of do a little thank you to the plants that they put all this work in to capture the sunlight so that we have a food supply. Even if you're not eating any plants, if you're against vegetables, there will be some animals that ate those vegetables that you then eat. So it's all part of the food chain. Okay. I believe I don't have any more slides. Is that true? Let's see. Dramatic. Nope. We're good. I will stop recording. I will stop annotating. Sorry, this will take me a while. Stop recording. Stop sharing the screen.